At the conference, we're going to present our very recent work on pharmacogenomics, which is a study of inherited basis of intravidual differences in drug response. So the pharmacogenomic study in our lab was focused on finding some genetic basis of our CML patients that may be associated with imatinib response. In this work, we were working in CML patients who were treated with imatinib first line with the standard dose of imatinib. It means that the dose was not changed during the follow-up, which was first 12 months since imatinib initiation, because the 12 months landmark was the criteria for response evaluations. And we look at the single nucleotide polymorphisms in the sequences of promoters of genes that encoding drug transportations through the plasmatic membrane. And we selected 19 genes, and we performed high-throughput single nucleotide polymorphism screening in the promoter sequences of those 19 genes encoding drug transporters. So, and among them, I suppose 5 of the transporters are or have annotated function of imatinib transportation.
Okay. From the abstract, I saw that the SLC and ABC genes were especially included. Could you tell us more about those as their role in healthy cells and how that changes in CML?
So we see clearly from our results significant differences in the frequency of allele of identified SNPs in promoters of SLC22A4 and SLC22A5 between optimally responding patients to imatinib and non-optimally responding patients to imatinib. And comparing the allele frequency of those SNPs, identified SNPs, to European population, I mean SNPs frequency comparison between optimally responding patients, non-optimally responding patients with European population, we clearly saw the shift in allele frequencies in non-optimally responding patients in comparison to European population. We believe that those SNPs may be a candidate to be implemented in real clinical practice. But before that, we need to prove this on really large cohort of CML patients. Because for such association studies, it's really good to start with a cohort of, let's say, 500 individuals, which is always complicated when we're working with rare disease like chronic myeloid leukemia is. So we believe we will have a nicely international collaboration on that to prove our observation on really large cohort of CML patients before we may implement these SNPs into a real clinical practice. But upon our genetic analysis, we perform linkage disequilibrium analysis with the unidentified SNPs, and we found that those SNPs are in highly significant linkage disequilibrium with other regulatory single nucleotide polymorphisms, which are located in non-coding intronic regions of both genes, which assume that our findings may have a regulative basis, and that the set of genotypes which is inherited for each patient may really matter or may result on what will be the expression of both genes, and this may impact the expression of the protein, the carriers, imatinib carriers, and finally this may result on bioavailability of imatinib. And we know that sublethal intracellular concentration of imatinib may cause resistance development in leukemic cells.